I guess we could move on to our next speaker. I would like to welcome Eapsa Berry. Eapsa is currently a postdoc at the Delhi University, and she applies phylogenetic methods, plant morphological principles, and carries out ecological studies to deal with some emerging evolutionary questions. Eapsa will speak today on petal folds in some funneliform flowers and the hierarchical surface microstructure in their corolla. So I would leave the time to Eapsa.
Thank you. Is my slide visible?
Yes.
Okay. Namaskar, everyone. Shall I start?
Please go ahead.
Namaskar, everyone. Today's discussion would be about folds and petals and their hierarchical surface microstructure formed by wax depositions. I'm not able to move my slide. Maybe now. Yes. We would basically talk about funneliform flowers of Ruellia, some unique features of the petal surface and its folds and wax composition therein. We would also discuss functionality and ecological implications of this trait. Finally, there will be a brief mention of distribution of funneliform flowers across the phylogeny of flowering plants. Why are studies like this important? Flower shape is a commonly studied attribute of floral architecture. Selection pressure comes through a combination of floral traits that have shown their presence for and presence by a particular type of preference for and preference by a particular type of pollinator. For example, our evolutionary studies in rhododendron showed us that multiple independent occurrences of different species with a common set of combination of floral traits were predominantly pollinated by a particular group of pollinator, thus forming an association between pollinator group and a trait group, technically called a pollination syndrome. So across the diversity of flowering plants, we have looked for funneliform flowers. Funneliform flowers are known to be a derived trait in angiosperms, and thus we would concentrate on this highly diverse group of asterids, clade of asterids, and Acanthesi to be particular, and Ruellia, the genus within Acanthesi, which falls under Lamids. Acanthesi is a species-rich tropical family found in the Indian subcontinent, and particularly Ruellia, which is the second largest genus in the family with around 300 species. They have funneliform flowers with petal folds and are now widely grown in urban areas in Delhi. We are interested in flowers which have fused petals that gradually spread or open out distally, forming a cone like a funnel. There are other fused flowers, flowers with fused petals. Some examples are given below. Here is a striking example. Here are some examples of funneliform flowers with petal folds, and just to show an exception, a pitcher-shaped primula flower without petal fold. Please note here that we have called the fold that bulge away from the central axis of the flower as an outfold, and the one towards the central axis of the flower as infold. We come to an important clade accounting for a majority of funneliform flowers with petal folds that we have observed, asterids. Datura from Solanacea shows a pattern of presence of folds in the petal. Geigelia has crumpled petals. Gentiana again has folds on the petal throat. Ruellia has folds on the petal throat. Ipomia shows this, you can see the color variation, but there are these folds here. And rhododendron also has funneliform flowers and folds of a different kind, but yes, there are these examples. Funneliform flowers add to volume of the petal and corolla for good reason, as shown through these examples of fabric manipulation in dresses that lead to volume. In order to study the petal tissue, we studied the surface of funneliform flowers with petals, their surface microstructure and wax depositions on them. Numerous functions of wax deposition have been reported and newer functions are being studied. They inhibit the loss of water, they provide a hydrophobic surface to the petals, they protect the petal surface from UV, they emit volatiles and to help attract pollinators, and through optical effects to attract pollinators, and to provide grip to the pollinator, among other constantly studied functions of waxes on petals. We now first move to looking at the petal surface of Ruellia. These are the various types of cells found on the floral epidermis, and it is known that petal epidermises have secondary depositions too. The surface map shows conical and knobby palleted cells on the lobe region. And as we move from the distal to the proximal side of the petal, we find that the cells get flatter and flatter, and the throat region has cuticular secondary depositions, which are denser. This apparent difference in lobe and throat region that bears the folds, infolds, and the outfolds made us want to see the composition of wax components. And we studied the fatty acid composition in wax on both the infold, both the lobe and the throat. Eventually, we will study all the different components of wax, the alkanes, the aldehydes, the ketones, alcohols, and ethers and esters. It is interesting to note the substantial difference in fatty acid percentage composition between the lobe region and the throat region that bears the infolds and the outfolds. You can see the higher percentage of saturated fatty acids in the outfold, in the throat region that bears the infolds and the outfold compared to the lobe. And as saturated fatty acids are known to increase rigidity, the throat is sturdier compared to the lobe of the petal. Also, we note the presence of very long chain fatty acids, which is also known to be a desired property of the petal. From this idea that the throat is different in wax composition from the lobe region, we move on to study the infold versus the outfold in the throat. The scanning electron micrographic studies show that the outfold has denser cuticular depositions, primary and hierarchical, which is secondary depositions, compared to the infold region. You can see these having more compact and denser patterning of the wax. We then move further to see whether the two folds, infold and outfold, weigh differently. This would allow us to study the relative wax load in our ongoing analyses. And we empirically measured the infold part and the outfold part from a population of approximately 30 to 40 plants. The part cut out were approximately 1 centimeter long and 2 millimeter wide. And we did a number of multiple sampling, and we found that indeed, statistically, the outfold weighs more than the infold in both the fresh and dry weights. Now we move on to understand functionality of these petal folds and also to scientifically speculating about them. Flowers that get folded or swamped with rainwater may have lower fitness due to reduced pollen transfer or pollen visitation. You can look at this video here. We can find the scene after the rain. The throat gets water in the rain, and when you squeeze the throat gently, the water comes out through the outfold. The cuticular depositions are hydrophobic in nature, and thus they protect the flower or sort of enhance their longevity. Secondly, the throat are, they sort of appear like better mechanical support as a corrugated asbestos or thin sheet, and the cuticular nanoridges on these infolds and outfolds work like the ribs of the ribbed iron rods. Thirdly, bacterial growth is found to be checked by cuticular depositions. So cuticular depositions, wax depositions, epicuticle and cuticular both are found to have been shown to check or avoid bacterial biofilms growing on these surfaces. And last but not the least, the pollinator, as you see in these photographs of Ruellia, they land on the flower, and the flower petal gets a proper the pollinator gets a proper landing on the flower. Finally, we move on to see the presence and distribution of funneliform flowers with petal folds across the phylogeny of angiosperms. The highly diverse clade of Asterids, which is species rich and is, shows presence of, high numbers of funneliform flowers. We used Floragator that has an online key to morphological traits in flowering plants with 200 documented families. Approximately 400 families are there in angiosperm. So among those 200 families, data extracted from Floragator tells us that out of 200 families, there are 17 representative families which belong to Asterids, caryophyllales, and monocots. And among those 17, 15 belong to Asterids. Within Asterids, out of the 15 families, 7 families show presence of petal folds in funneliform flowers that we have talked about. We observed these families and orders in these families, the families in these orders, and we found that there are multiple genera that show, multiple occurrences on this evolutionary tree, which suggests that there is a functional association of these folds with types of floral forms. Similarly, we see this in Acanthaceae, where there are multiple genera that show petal folds, including our species of study, Ruellia. Some questions remain. Do all folds and petals have similar definable functions? Ontogenetically, how petal folds develop, whether there is possibility of developmental constraint. Petal folding is not necessary condition for the funneliform. There are exceptions and deviations. For example, in Alamanda, there are no folds, but a considerable thickened throat region is present. And in Rhododendron Dalhousie, there is only infold and no outfold. For this, firstly, I would like to thank my teacher, constant collaborator, and PhD advisor, Professor Petal. Then my collaborators in the analysis and more about wax composition, Professor Mishra and Dr. Ashish. Then my present and former postdoc mentors for continued guidance and support, Professor Tandon, Professor Pandit, and Professor Olson. And finally, the Society for the Study of Evolution and the Indian Society for Evolutionary Biologists for giving me this unique opportunity. Thank you all for your kind and patient listening. I would be happy and grateful to take questions and comments from the audience.
Thank you, Eapsa. Very interesting work.
Thank you.
Yeah, before letting other people throw the questions in the chat box, I have a quick question. Just curious about the what is the changes across the time for the wax composition in the petals? Like, let me rephrase myself. Do the wax composition in the petals will be like change across the time, like from the bud of a flower to almost dying, drying out? Will that be changing?
Yes, I think I have some images of, I haven't shown those, like some images of developing flowers. So you can, of course, I haven't done the composition analysis as you're mentioning, but the photographs show that the depositions and the secondary hierarchical depositions change over time. Now, given that, that means the sort of wax load would change. Again, as you see that the flower has its course, like there are two phases mainly, the cell dividing phase and the cell elongation phase. In the dividing phase, the cells are more producing, they are sort of very small. If you look at the microscopic cells, they're very small, they are dividing, and they are filling up that sort of in that little area, they are all sort of coming up. And then when you have this phase when it is expanding, then each cell has they're taking their own shape and they're sort of getting structure. Now, there have been studies and other, I think one study is about from the group of Beverly Clover, where they've shown that, this surface cuticular deposition expansion leads to the overall deposition change. At the same time, I'm saying now we are looking at sturdiness and presence of saturated and unsaturated fatty acid. Other components too have equal roles to play. And all of this shows a dynamic picture of developmental phases of the flower.
Thank you. We have a question from the audience. If different families are showing different form of petal folds, is there any correlation between petal folds and pollinator preference?
This is an interesting question. Like the last slide where I talked about the question, that still many things remain to be understood. But what we see is, like in the case of the rhododendron petal fold, there are only infolds, and that infolds are, when you visualize it at the microscopic level, or through naked eyes, because these are big flowers, they are on the regions where the nectaries can be. So they might be performing different functions. There is a larger set of functions that we can say that, mechanical strength and those things are common, but pollinator preference is one thing which will be related with many other. It's a complex combination of traits that interacts with the pollen, and of course this needs to be studied. It's interesting.
Thank you.
Thank you.
Let's see, do we have other questions? Okay, I guess that's it. Thanks, Eapsa.
Thank you. Namaskar.
Yeah. So now we will have an hour break, and we will hopefully meet everybody again around 1:30 p.m. Indian time, which is 13:30 Indian time. So for the last session, for the next session, when we come back again, we're going to use a different Zoom link. So now we will drop another Zoom link, and with the meeting ID, with the password in the chat box. Also, please check your emails for the link too. We're going to send to you, or we have sent to you. So please copy the Zoom link and the password with you. We will see you later for the last plenary talk and a group discussion. Just a reminder, please don't share the information of the Zoom link and password on social media, please. And if you have any question before you leave here, please submit a question to the Q&A box, and our speakers will check and communicate with you. Thanks. Thanks for attending this morning session.